We all know that plants make their energy from the sun. That's kind of the main concept of photophosphorylation. But to get a closer look at how plants actually work, we gotta look at the two specific parts of photosynthesis. The first one we're gonna look at is the light-dependent reactions. Okay, now this is one of the parts of the light-dependent reactions. It's called photosystem 2. This occurs in the thylakoid membrane. What happens is the sun is constantly releasing photons of light. One of these photons is absorbed by a chlorophyll molecule. Then two of these molecules are so excited that they have a proton of light that they both release two electrons, and they are picked up by the acceptor, piquinone. And now the piquinones are reduced because they have two electrons each. Another part of the light-dependent reactions is photolysis. This part is constantly ongoing, and the water is basically split up into each of its parts. There is the hydrogen atoms, which are used later. We'll talk about that. The electrons, which can be recycled and also used again for photosystem 2. And then the oxygen is a waste product, which is basically pooped out by the cell, releasing to the atmosphere outside the cell, or underwater if, we got an aqueous plant. Next part, electron transport chain. The big wigs of this part are the electron carriers, which are NADP+ and ferredoxin. We'll talk about ferredoxin later in photosystem part 1. But for now, we're mostly going to talk about NADP+. Now, the proton gradient. Remember those hydrogen protons from earlier? Yeah, those are important. So, what happens is, also, the piquinone from earlier is also very important. So, inside the thylakoid, there's a high concentration of those hydrogens from earlier, and outside there's a low concentration. So basically what happens is the piquinone from earlier is going to transfer its electrons all over, and it'll pump protons across the membrane space, the yellow part, into the thylakoid. So once this develops, like this concentration that we have here with the large amount on the inside, smaller amount on the outside, not necessarily amount, concentration, this is how we generate ATP. ATP synthase uses this proton gradient to make ATP. The protons move from the high area inside the thylakoid space to the area of low concentration outside, so eventually the inside is going to move to the outside. And then the energy is going to be released by the protons passing through, and that's going to make our ATP, baby. This is also called chemiosmosis because it's where protons move through a membrane. Now time for photosystem 1. And this kind of goes hand in hand with NADP+ reduction. So I'll talk about them in a minute. So first, you got the plastocyanine from earlier that was transformed using the electron transport chain. And this is going to go through photosystem 1, basically with more chlorophyll molecules, where more light comes through. Basically the same thing. And it's going to turn into, guess what? Ferredoxin, that important proton carrier from earlier that I talked about. And then the NADP+ and a hydrogen and two electrons, guess what they're going to make? They're all the way out there. NADP reductase is going to turn all of those things that are just floating around, having a good time in there, into NADPH. Bam. But sometimes there's not enough NADP+. And so when this happens, a bunch of the happy little electrons floating outside, they're going to return to the electron transport chain. You can see this big gray arrow, so that they can pump more protons, which will get more ATP production, and therefore produce more NADP+. Wow, that was crazy. Now we have moved on to the light independent reactions. And we have also graduated out of the thylakoid membrane and thylakoid into the stroma. Congratulations. The first phase of the light independent reaction is carbon fixation. Carbon fixation is where a carbon dioxide and a ribulose biphosphate are fixed by the enzyme Rubisco. And they make 2 molecules of glycerate triphosphate. 2 molecules. That's important because the whole thing kind of gets scaled up and confusing. But don't worry. Phase 2, reduction. In reduction, we have the 2 triphosphoglycerates from earlier. And guess what? We got the ATP from the light-dependent reaction and the NADPH from the light-dependent reactions. Excuse me. And guess what all of this can possibly make? Triglyceridyl triphosphate. Isn't that crazy? And this could possibly turn into an organic compound, such as glucose. So, remember, it starts out as 2 triphosphoglycerates, and then it turns into 6 glyceraldehyde triphosphates. That's important to know. And then one of those can turn into an organic compound, such as glucose. Phase 3, regeneration. So remember how I said those numbers were important. 1 out. It's glucose or whatever it wants to be. Those other 5, they are recycled. We recycle here. We are environmentally conscious. Into ribulose biphosphate. Wait, doesn't that sound familiar? Yes, it does, because that was in the carbon fixation. Isn't that bonkers? So, it can either be turned into glucose, basically, or back into ribulose biphosphate. That's how plants make their sugar. Oh,